Quickly, guys, we need to start. We're gonna be late for the lunch break. People outside, come on. Maybe I should start the attendance and everyone will be here, right? But they will soon leave then, that's the problem. Maybe I should take 2 attendance, one at the beginning and one at the end. What do you think? Good idea? Sorry? Okay, so today we are supposed to start with the glycolysis, but I thought before we start the glycolysis, we need to review and get familiar with the important concept about metabolism. Okay, so this is a glimpse of the different metabolic pathways that we will be studying in Mol 1 and Mol 2. Please take your seat. Whoever is standing there at the end, please take your seat. Group leader, do you want to record the session? It is recording? Okay. So today we're going to review the metabolic pathways. So what are metabolic pathways and how these pathways are regulated? And what is the definition of all these terms in biochemistry? What's feedback inhibition? What's regulated step, committed step, rate limiting step? And what is homeostasis for the cell and how it is maintained? And when things go wrong because of genetic mutation, what are these inborn error of metabolism? So first of all, let us review, just to make sure you are all familiar. The material inside the cell, we have the large molecules or the biomolecules. We have 4 types of large molecules. What are these 4 types? Who can remind us? What are the 4 types of large molecules or macromolecules inside the cells? Yes? Carbohydrates, proteins, lipids, and nucleic acids. So these 4 macromolecules, they are the, if you want to say, the ingredients of our metabolism. Okay, because things are made out of them, and these ingredients, they will be degraded or catabolized, as we will see. What are the metabolic activity of the cells? So inside our cells, we have metabolic activities. So all the reactions taking place inside the cells, they are defined as metabolic activities of the cells. The cell needs to do 2 things. The cell needs to make or synthesize the large macromolecules that are needed for the cell survival. Okay, so these are what we call, so when we talk about making or synthesizing things, this is the anabolic pathways. Okay, so in anabolic pathways, we are making large, complex molecules from smaller ones. So we could be making proteins from amino acids, we could be making complex carbohydrates from small sugar, etc. So the cell, to survive, needs to make proteins, and these proteins are made from amino acids. The cell needs to make complex carbohydrates from monosaccharides. The cell needs to make membrane lipids from fatty acids and other molecules. And the cell needs, of course, to make nucleic acid, whether it's DNA and RNA, and they are made from nucleotides. So this is one part of the metabolic activity of the cells, to make things which are needed for the cell survival. So anything, when we talk about synthesis, any biosynthetic pathway, we call it what? Anabolic. Okay, so this is like one part of metabolism, which is anabolism, making large molecules from smaller ones. All anabolic pathways will require input of energy. So we need to invest in energy, we need to put energy in, and this energy is required in which form? In the form of ATP. So ATP is like the energy currency of the cell. Okay, so here in Riyadh we use Riyal, right, to buy things. The cell, to make things, they need ATP. So we said anabolism is needed to make large molecules from smaller one, but what is the other part of the metabolic reactions that take place inside the cell? These are the catabolic pathways. So what happens in catabolic pathways? The cell will break large molecules into smaller ones in order to generate what? Energy. Okay, so the cell has to generate energy, and how the cell will obtain energy? We're not going to feed the cell with a spoon, no. Nutrients will be oxidized to supply ATP. Why the cell need energy? Why the cell need ATP? To do what? For biosynthesis, to make things. This is one. What else? Why a cell will need energy? Yes? Okay, so if it is a muscular, if it's a muscle cells, for the contraction, we need ATP. What else? Why the cell need energy? Yes? Sorry? Active transport. Yes, so we need to maintain all these ion pumps. What else? Yes? Yes, so she mentioned the contraction or motility. Yes, so biosynthesis, this is when the cell needs to make large molecules like membrane lipids, etc. The cell needs this energy for active membrane transport, for motility, and for contraction if it is a muscle cells. Which of the following is the most important factor for cell survival? Which out of these four is the most important for cell survival? If we have deficiency in ATP, which of these four must be met for the cell to stay alive? Is it biosynthesis? Yes. So who says it is membrane transport, active transport? Okay, good. Why it is membrane transport is the most important thing for the cell to stay alive? Yes. Because ion concentrations need to maintain. If we don't maintain this balance of ions, what will happen? Yes? The cell will die. Why? What caused the death of the cell in that case? Yes? Osmotic pressure, right? Yes, so these processes, they're all catabolic, and the most important one, the top priority for the cell is active membrane transport, as we will see later, because without proper, gradients of ion, the osmotic pressure will be altered and the cell will die. So all of these four are needed they require energy. And where do we get the energy? By breaking things. And when we break things or we degrade things, this is called catabolism. Okay, so catabolism is the degradation of large molecules into smaller ones. You need to remember two things in biochemistry or in our metabolism. So we have catabolism and anabolism, but these two must be balanced. Okay, so there must be balance. So things should not be going fast in one direction, but not too fast in the other direction. So we need to have a balance so that we maintain homeostasis. So we need to keep those molecules at a constant level. So the rate of anabolism must match the rate of catabolism. So there should be a balance. So we should balance the biosynthesis of molecule with their degradation. And you should also remember another factor, that different nutrients can be interconverted. What do we mean by interconverted? Yes, so in other words here, I'll give you an example. That's why I put this image here. So we can use glucose to make amino acids, and amino acids can be used to make glucose. Okay, so we're going from one type of molecule to another. This is how we, what we mean by interconversion of different nutrients. Okay, so we can use glucose to make amino acids, we can use amino acids to make glucose. This is the interconversion of the nutrients. Also, glucose can be converted to fatty acids. So also this happens inside the liver. Glucose can be converted. If we have too much glucose, excess glucose will be converted to fatty acid, as we'll see later in mole 2. In this diagram here, we are showing that cells use the nutrient for one aim, which is to make the large molecules to maintain, the function of the cell, but also the cell will use the nutrients in order to generate ATP. Okay, so these are the catabolic pathways. Things are being broken down to CO2 and water. So this is the ultimate fate of all the nutrients that come from the diet. Okay, so we eat, things go in the digestive system, then in the blood, and inside the cell only the small molecules, so glucose, amino acids, fatty acids are taken inside the cell, and they will have two fates inside the cell. Either they are used to make large macromolecules to maintain the function of the cell, or they are broken down in order to generate ATP. So they will be broken down into CO2 and water. Okay, so this is the fate of the nutrients and how inside the cell all the macromolecules are handled. Okay, so we need to stay alive, we need to eat. The food will be digested, the nutrients will be absorbed at that small level inside the cell. Inside the cell could use them to make, important molecules to keep the cell alive, or the cell will use them to make energy. And these molecules will be broken down to CO2 and water, generating lots of ATP. Now I have a question. Can cells choose their source of energy? So we have three sources of energy, right? So we have proteins, we have lipids, and we have glucose, right? So can a cell choose which one of these energy sources to use? Yes? Depending on its action. Okay, so for most of the cells, they have options. So they will have supply of energy in different forms, which is glucose mainly and fatty acids, because eventually the proteins will be broken down to amino acids, and amino acids are used to either make glucose or to make fatty acids. But not all cells have this option to choose which energy source they can use. Some cells will depend only on glucose. Can you give me an example of cells that depend only on glucose? Why brain cells will depend on glucose? Because? Yes? Sorry? Okay, so brain cells, their primary source of energy is glucose, but we will learn in mode 2 that, if we have starvation, they can switch to using ketone bodies. Okay, we're going to learn this in mode 2. Now we still don't know what is ketone bodies, but brain really need a continuous supply of glucose, but in starvation, they can switch to another mode of energy source. But I know one cell type that needs glucose, and we can only use glucose as a source of energy in all situations. Which cell type? Red blood cells. Why red blood cells can only use glucose as a source of energy? They cannot use any other thing. because they don't have a mitochondria, okay? And the other source of energy to be broken down, they need the mitochondria, but only in the cytoplasm, which is, the red blood cells, they only have a cytoplasm. We can do what we call anaerobic glycolysis, which will produce ATP, which is the only source of energy for red blood cells. Okay, so most cells can choose different substrates, so whether they can depend on glucose, fatty acids, or amino acids, but neurons and red blood cells, they are very strict with their energy source. So neurons will depend mainly on glucose, but as I said, in starvation, they can switch to something else. But red blood cells depend exclusively on glucose, so they can only use glucose for energy. So something to remember when we study the metabolism of these cells. So we learned that metabolism is divided into two parts. We have anabolism and catabolism. We said that in anabolism, the cell is making important molecules. In catabolism, the cells are breaking the nutrients in order to generate energy. Energy is needed for different purposes. The most important thing is maintaining the transport of ions. We learned that some cells depend only on glucose, like red blood cells. Now we need to know about the compartmentalization. What is the word here meaning? So we have the word compartment. What do we mean by compartment? What does this sentence mean? Metabolic processes are compartmentalized. Sorry? Classified? Yes. I can't hear because of your mask. Yes. Excellent. So your colleague is correctly saying that different reactions or different metabolic pathways will take place in different compartments of the cells. When we talk about compartment of the cells, you took a lot of cell biology in foundation. What are the different compartments? Do we have rooms, floors? What do we have inside the cells? We have the organelles, right? So we have the cytoplasm, and then we have inside the cytoplasm different organelles. So we have lysosome, peroxisome, the mitochondria, right? So these are the different compartments inside the cell. So when I say metabolic processes or processes are compartmentalized, it means that each metabolic pathway has a specific location inside the cell. That's why we said like red blood cells don't have mitochondria, therefore they can only depend on glucose, because things happen in the mitochondria, they don't happen anywhere else inside the cell. So usually cytoplasm contains biosynthetic pathways. So what do I mean by biosynthetic pathways? We are making large molecules. So whenever there is a synthesis of something, this is made in the cytoplasm. And also we have a few non-oxidative catabolic pathways. What do we mean by non-oxidative? They don't involve oxidation reduction, and we'll talk about this later next week, inshallah. And also in the cytoplasm, this is where we store large molecules such as glycogen and fat. Okay, so this is inside the cytoplasm. So biosynthesis, non-oxidative pathways, and storage of large macromolecules. You learned a lot about the ER and the Golgi. Why we need the ER and the Golgi? For the processing of the proteins, right? So all the post-translational modifications of proteins and also for membrane lipids that take place in the ER and the Golgi apparatus. And then the lysosome, what do we have inside the lysosome? We have enzymes that break molecules, so we call them hydrolytic enzymes. And these are used to degrade large molecules. And also we have the peroxisome. In the peroxisome, this is important for mol 2, we have some reactions for the lipid metabolism. So everything happens in its own place. And most importantly, the mitochondria. The mitochondria is usually described as the powerhouse of the cell because most of ATP, or 90% of the ATP, is produced in the mitochondria by the oxidative phosphorylation that we will study next week. So red blood cells don't have the mitochondria, they don't have oxidative phosphorylation, and they depend on glucose only to generate ATP by anaerobic glycolysis, as we will see later. Okay, so these are the different compartments of the cells, and this is where the different reactions will take place. So some in the cytoplasm, some in the mitochondria, some in the lysosome, and some in the peroxisome. Okay, when we talk about fuel oxidation or the breakdown of fuel, we need to remember that we need oxygen, and this is why, without oxygen we cannot stay alive, because we have what we call cellular respiration. When the cell is respiring, it is using the energy from the nutrients, and it's using oxygen to break down these large molecules down to what? CO2 and water, and therefore generating lots of ATP. So this is fuel oxidation. This is for glucose. So glucose in the presence of oxygen is metabolized or broken down to CO2 and water, generating lots of ATP. And this doesn't happen in one reaction. This happens in multiple pathways that we are going to study in Mol 1. And here we have the breakdown of lipids. So this is a fatty acid which is be broken down again in the presence of oxygen to CO2 and water, and you will study this in Mol 2. Okay, so oxidation is basically here, when we talk about fuel oxidation, we are talking about the catabolism of nutrient, or the breakdown of nutrients. So if it is a glucose or if it is a fatty acid, they all have the same fate. They will be broken down in the presence of oxygen to CO2 and water in order to generate lots of ATP. And for the glucose, this is cellular respiration, and it happens in multiple pathways. It doesn't happen in one reaction. And this is the beta oxidation of fatty acid that you will study in Mol 2. Yes, because glucose and fatty acid, they are type of fuel. Okay, the metabolic fuel for the cell. Okay, the fuel for the car is just, the gas that you put in your car. The fuel for the cell is the glucose and the fatty acids. Okay. This reaction is actually, a summary of multiple things. Okay, so here, like glucose, proteins will be broken down to amino acids, and amino acids will give rise to glucose. But at the level of the cell, it is glucose that's going to be oxidized into CO2 and water. You will understand the full picture as we go along in the course. Okay, so you need to understand that these complex metabolic reactions, like the oxidation of glucose that I have just mentioned, or even the synthesis of glucose, they don't occur in one step. It's not like your high school chemistry. You have one reaction. No. In metabolism, you have a series of reactions, or what we call a sequence of reactions. So these series or sequence of reaction, they form what we call metabolic pathway. So what do we mean by pathway in biochemistry? So this is a pathway. This is here a glucose being broken down into pyruvate, and lactate. So this is the first step actually of cellular respiration. So the first step for those who took A level, they know that in cellular respiration, the first step is glycolysis, right? So this is just the first step or the first stage, if you wish, of cellular respiration, which is glycolysis. What is glycolysis? It's a breakdown of glucose into pyruvate in the presence of oxygen. In the absence of oxygen, glucose is broken down to lactate. So this is not one reaction, this is a series or a sequence of reaction, and we call this a pathway. How are the reactions in a pathway linked together? If you see, this is the substrate. Okay, so in biochemistry, the reactant is called the substrate, and this is the product, right? So in the pathway, the product of the first reaction will become the substrate of the following reaction. And this is how they are connected, and so on and so forth. So in each step, the product will become the substrate. So the product will become a substrate, and this is how they are connected in a series of reactions or a metabolic pathway. Right. As you can see here, these reactions, they have two arrows. What do we mean by two arrows? So you have reactions with two arrows and you have reactions with one arrow. What? Yes? Yes, reversible and irreversible. So when I say reversible, it means that the reaction can go in both directions, in the forward and in the backward direction. Okay, if it is one arrow only, then it is irreversible. So we learned what is a pathway, and we said that the pathway can be a series of steps, and these steps can be reversible or irreversible. But these pathways, they have to be regulated. Okay, they just don't occur like this. They have to be under strict regulation. Why they need to be regulated? Because our cells need to adjust their metabolic activities according to the need of the cell and need of the body. Okay, so things don't just like happen like this. So every reaction in our body need to go fast or slow depending on the need of the cell itself and the need of the body. So these two things will control the reactions in our body. So cells have to respond to their own energy status. So if the cell has very little energy, then it needs to speed up the reactions that will produce energy, and also the availability of the nutrients. So our metabolism doesn't happen the same way whether we are eating or whether we are fasting. Okay, so if we are eating, some, like after a carbohydrate-rich meal, some pathways will go fast and some pathway will go slow. The same thing, if we're fasting, some pathways will be activated and some pathway will be inhibited. So energy status, availability of nutrients, and hormones. These are the three regulatory keys, if you want, to our metabolism. So how do we regulate things? We just tell this pathway like you go fast or this pathway you go slow. So how do we convey this information? How do we make things happen faster or happen slower? By controlling what? What do we control? We regulate the enzymes, yes. So we can make a pathway go faster or go slower by regulating the rate of the enzyme or by controlling the enzyme. How can we control the enzyme? There is two ways of controlling the enzyme. There is a fast way and there is a slow way. The slow way is by adjusting the amount of enzyme. So we make the cell express more of this enzyme. So we enhance the gene expression of this enzyme. Or we can make this enzyme not be degraded. So we can control how much enzyme we are making or we can control the degradation of the enzyme, but this is slow regulation. So we can change the rate of synthesis or degradation of the enzyme, because these enzymes, they have a certain lifespan, so it could be 1 hour or several days. We can control degradation or we can control the synthesis. This is called long-term regulation of enzyme activity. But the rapid one, the fast regulation of enzyme activity, is by covalent regulation. How do we regulate the enzymes by covalent regulation? By adding or remove a phosphate group to the enzyme. So enzymes can be phosphorylated or dephosphorylated. So this is reversible. So do phosphorylation make the enzyme less or more active? Excellent. So it depends. Some enzymes will be active when they are phosphorylated, and some will be active when they are dephosphorylated. Okay, so it depends on the enzyme itself. There is no rule, but it depends on the enzyme. So to make the enzyme phosphorylated, you need another enzyme, and this other enzyme called protein kinase. What does protein kinase do? It will add the phosphate group to the enzyme, making it more or less active. To remove the phosphate group, you need the phosphatase enzyme. So what do the phosphatase do? It will remove the phosphate group, again making the enzyme less or more active. So how these phosphatases and kinases regulated? Again, they are regulated by nutrients, availability of metabolites or hormones, as we will learn later in the course. Okay, so this is the enzyme that we're trying to regulate. This is the phosphorylated form of the enzyme. We could add a phosphate group from ATP, so ATP will become ADP, and the enzyme will be phosphorylated, but we need the kinase enzyme. So kinase add the phosphate group, and the phosphatase will remove the phosphate group in the form of inorganic phosphate. So this is one of the two fast types. So we said we have slow regulation by making more or less of the enzyme. We have the fast regulation by phosphorylation, dephosphorylation, but there is also another fast regulation, which is allosteric regulation. Did we come across the word allosteric before? What does an allosteric regulator mean? Which lecture we came across the word allosteric regulator? Globular protein, thank you. Yes, when we studied the globular protein, we studied the definition of allosteric regulator. What does the word allosteric itself mean? Allosteric is another site, okay? So allosteric regulation or an allosteric regulator will bind to one side of the protein or the enzyme, and it will change its activity at another site. This is what we mean by allosteric regulator. And when we say change the activity, does it make the enzyme active or less active? It depends. So allosteric regulators, they can be inhibitors, or they can be, what's the opposite of inhibitor? Activators. Okay, so under allosteric regulation, we have activation and we have inhibition. Okay, so this molecule, which is the allosteric molecule, will bind at the site other than the active site and will influence the activity of the enzyme. So this allosteric effector could be a substrate, intermediate, or a product of a pathway. A common allosteric effector is ATP. So ATP, the molecule, the ATP itself, can be an allosteric regulator of many enzymes in the metabolism. So we define that effectors can be activators, so they increase the enzyme activity, or they can be inhibitors that decrease the enzyme activity. And allosteric regulations can be in two major forms. So we can have feedback or feed forward. So what do we mean by feedback and feed forward? Let's see the example here. First, let us play this video together. Can you hear anything? No. Okay, you can play this video at home. No time to waste. Let's look at the diagram together. So this is an example of a metabolic pathway. We have a series of reactions. So we started with the substrate A and we end up with the product E. If inside the cell E starts to accumulate, then E itself can switch off this pathway by inhibiting the enzyme of the first step. This is a typical, control mechanism inside the cell. So if we are trying to make a certain molecule, and this molecule starts to accumulate, then the product of this pathway will inhibit a reaction that happened earlier in the pathway. And usually, this reaction would be at the very beginning of the pathway, and we'll explain why later. So this type of regulation, we call it feedback inhibition. Why inhibition? Obviously, because things are being switched off, or that we are trying to inhibit this pathway, as you can see from the negative sign. And why feedback? Because this molecule which is produced by this pathway, which is the consequence of this pathway itself, it acts as a regulation of an earlier reaction. So we call this feedback inhibition, and this is very common. Actually, most pathways inside the cell, they are regulated by feedback inhibition. And this is a type of allosteric regulation. So this is the definition of feedback inhibition. So a product which is formed later in the pathway acts as an inhibitor of an earlier reaction. So this pathway should be only active when E is needed. If we have too much of E, then we have to switch off this pathway. And this happened to most biosynthetic pathway inside the cell. There's no point of keep on making E if we have too much E inside the cell. And ATP is the most important feedback inhibitor in catabolic pathway. Okay, so in biosynthetic pathways, the feedback inhibitor is the molecule that we are trying to synthesize. But in catabolic pathway, the inhibitor of the molecule of the pathway is ATP. So if we have very little ATP, catabolic pathway will go faster. If we have too much ATP, then catabolic pathways will be inhibited. So this is an example of feedback inhibition. You should also remember when we talk about level of ATP. So ATP is the energy currency of the cell. If we have too much ATP, it will signal high level of energy in the cell. ATP is made from ADP and AMP. So if we have too much of ADP and AMP, this will signal what? High level or low level of energy in the cell? Low level, okay? So you should always think of ATP. Too much ATP is we have high energy level. Too much ADP or AMP, it means we have low level of energy inside the cell. So these two are inversely related. When one thing go up, the other will go down. So again, this is very important because for most pathway actually for all pathways, if ATP will inhibit, then ADP and AMP will activate. So they will have opposing effect because ATP is signaling high energy level and ADP and AMP, they are signaling low energy level. So they always have opposite effect in regulation of our pathway. Okay, so here again, we see the feedback inhibition. But another type of regulation is the feed-forward regulation. What do we mean by feed-forward regulation? So in this case, it's the opposite of feedback. Feed-forward means if we have accumulation of B in this pathway, it will make reaction E4 or the enzyme E4 go faster. Okay, so a reaction that happened early in the pathway and produce a product B will make a reaction that happened later in the pathway go faster. Okay, so this is an example of feed-forward regulation that we will see in later lectures. Now we need to know the different terminology that we use in the regulated step. So we said we need to regulate enzyme. We can regulate enzyme by different mechanism. The fast way is by either phosphorylation, dephosphorylation, or by allosteric regulation. We mentioned the feedback inhibition as an example of allosteric regulation. Now let us focus on the different steps in a pathway and how we describe them. What are the different terminologies that we use to describe the different steps in a pathway? Okay, so when we want to regulate a pathway, do we regulate each step of the pathway or we regulate certain step only? Yes, so we cannot regulate all the reactions. We have to regulate only certain reactions, and these reactions which can be regulated, we call them regulated step. And there are certain criteria for regulated step. A regulated enzyme must be present in lower activity than other enzymes in the pathway. What does it mean by lower activity? So this enzyme, usually the regulated enzyme, is usually an enzyme that is slow. It has, you will learn this in Mol 2 about Vmax and Km, etc. But a regulated enzyme usually are the ones that have a very slow activity. And that's why we call them this regulated enzyme with a slow activity. It will catalyze the rate limiting step. Okay, what do we mean by rate limiting step? So it is the slowest step in the pathway. Why it is the slowest step? Because the enzyme has very low activity. So this is one of the criteria of the regulated enzyme. It must catalyze the rate limiting step. It should only work in one pathway. So you cannot regulate the activity of an enzyme which is used in different pathways. So the enzyme that you need to regulate should be an enzyme that is unique for a pathway, for a certain pathway. And it should catalyze the first irreversible reaction in the pathway, or what we call the committed step. Okay, so what is the committed step? It is the first irreversible reaction in the pathway. So this is the criteria. These are the criteria for the regulated step. 3 important criteria. It has to be a slow-acting enzyme, so it has to be the rate-limiting step. It has to function only in one pathway, and it should be the committed step of that pathway. Why we need to regulate only the committed step? Or why the regulated enzyme has to be the committed step? Because we don't want to accumulate other products which are not necessary. What does this mean? Okay, let me show you an example here. Okay, so here we start with 1, we could end up with 3 or 6. The question here is asking which of the following enzyme catalyze the committed step for the pathway leading to the formation of 6. Yes, so C is actually the enzyme catalyzing the committed step. Why C is the committed step? Because once I go down to here, I cannot change my mind and go back and do 3. Okay, so once I'm down this road, I am committed in making 6. So C is the committed step for the pathway of making 6. It's the first irreversible. Okay, so here we have two options. So that's why C is the first irreversible reaction, and it's going to commit the pathway into making 6. So for now, remember that these are the criteria for regulated step. It has to be the rate limiting, the slower enzyme. It has to be common for this pathway, or not common, unique for this pathway, and it has to catalyze the first irreversible reaction. So why here I control C and not D or E? Why can I not control E? Why the regulated step is not E or D? Sorry? D and E, they're not the first committed, okay? They're not the committed step, because if I switch off only E, then I will still have accumulation of 4 and 5. Okay, so we mentioned feedback inhibition. So if I have too much of 6, I want to switch off this pathway, I should switch off C. Why only C? Because if I switch off E, then I will still have accumulation of 4 and 5. And I don't want to accumulate 4 and 5 because it's going to be waste of ingredients to make 4 and 5. So I need to switch off things from the beginning, from the first committed step in the pathway. So that's why, to inhibit the formation of 6, I need to switch off here things at the very early in the pathway. Okay, you studied homeostasis. Who can define homeostasis for me? To maintain a constant internal environment. And this is usually applied for the body, right? So our body is always trying to maintain homeostasis, but the same applies to our cells. Our cells are also trying to maintain a constant internal environment. Okay, so homeostasis is internal stability, whether we're talking about an organism or whether we're talking about a cell. So we have why we need homeostasis: to preserve the cell, for the cell to stay alive. Okay, so we need to have an internal pools of certain nutrients and molecules inside the cell. So this is part of homeostasis. And also we need to respond to changes. So if the cell is exposed to heat or toxic substance, the cell needs to respond to these changes and to try and restore its internal stability despite all these changes. And the most important thing, as I mentioned earlier, is to maintain the ATP concentration. Why we need the ATP concentration? To drive the ion pumps. Without the ion pumps, the cell will die. But we also need the ATP to maintain biosynthesis, to maintain transport processes. So the maintenance of the ion gradient is the most critical function of ATP. Why is that the case? Because if we don't have enough ATP, the pumps will not work. As a result, there will be osmotic stress and the cell will die. And how the cell will adapt? How will the cell maintain its internal constant environment? If we have decline in the level of energy, what will happen? What will the cell do? It will adapt. And how will the cell adapt? Either by increasing transport of the nutrients, so it will increase the uptake of nutrients, or it will start breaking down any stored macromolecules. So if we have, let's say, stored glycogen, the cell will start breaking down that glycogen to generate energy, or it will start to activate any energy generating pathway. So this is one of the adaptive, this is examples of adaptive response. So if we have decline in energy level, there will be increased nutrient uptake, there will be a switch off of any catabolic, there will be switch on of the catabolic pathway. So we're going to activate energy producing pathway, and we are going to mobilize any stored macromolecules. And we will inhibit any biosynthetic pathway. This is in the response of decline in energy. So this is how the cell will adapt to a low level of energy. So what will happen also, the cell will go into a low energy mode. It's like when your phone is on the low battery, you can switch it to low energy mode. So the cell has a way of going to low energy mode, or even the body will go into low energy mode. So anything that consumes lots of energy will be slowed down. So the glucose synthesis in the liver will slow down. Our thinking will slow down, the muscle contraction will slow down. All these happen when we don't have too much energy. In order to direct the ATP for the survival processes, and what is the survival mechanism? The most important one is the ion transport. So this, what will happen in response to a decline in the level of energy. And the opposite will happen after a carbohydrate-rich meal. If we have too much energy, what happens if you are exposed, or our cells are exposed to too much energy? So glucose is actually a toxic substance. We should not keep that glucose at high level in the blood. Glucose should not accumulate in the blood because it's a poison if it is present at high level. Yes, we need the glucose to stay alive, but if we have a high level of glucose in the blood or hyperglycemia, this acts like a poison for our body. So what will happen after a carbohydrate-rich meal? We will try to lower down the level of glucose. So how do we lower the level of glucose in the blood? We start breaking that glucose, or we mobilize it to the liver to be stored as glycogen. So we do anything we can just to lower down the level of glucose. So we start actively breaking down that glucose, or we store it in the liver as glycogen, or we store it also in the liver as fat. So this is after a carbohydrate-rich meal, and the opposite happens during fasting. So when we're fasting, there is very little glucose in the blood, and some cells will switch from glucose oxidation to fat oxidation in order to save the glucose for the neurons, because the neurons, they need primarily the glucose, and other cells like the red blood cells that depend on glucose because they cannot oxidize fat. How are these pathways or all these modes of regulation coordinated? They are coordinated by hormones. So we'll study in mode 1 and 2 the hormones, the different hormones that regulate our pathway. We have 4 hormones. We have insulin and glucagon, and they are always having opposing effect. So insulin is released in our body after a carbohydrate-rich meal. So whenever we are eating, the pancreas will release insulin, and glucagon is released also by our pancreas when we are fasting. And these 2 hormones have opposing effect. Insulin is trying to bring the blood sugar down, and glucagon is trying to bring the blood sugar up. Okay, so these are the 2 hormones which are secreted by the pancreas, and they control the blood sugar. But we also have other hormones that control the blood sugar. Adrenaline and noradrenaline, they are secreted in response to stress or physical activity. Also, they are used to mobilize stored fat and glycogen. And cortisol is released during prolonged stress. So if we are experiencing too much stress, if our body is under continuous stress, cortisol is released and it promotes the synthesis of glucose. So these are the 4 important hormones that control our pathway. Okay, the last slide here is about the inherited enzyme deficiency. We started late, so I can go on for a couple of minutes. So we are going to revisit this many times, but in biochemistry, we are going to learn about inborn error of metabolism. What do we mean by inborn error of metabolism? So there is an error of metabolism that a child is born with because of a genetic mutation, and this mutation is affecting the enzyme, so the enzyme is not working. As a result, we are not making a certain product. So what will happen? There will be, in this case, if we take an example of albinism, so a person with albinism or an albino, they cannot make melanin, and as a result, because of the genetic mutation, so there is a missing enzyme that is needed to make melanin. They cannot make melanin. Instead, they will have other metabolites accumulating in the body. So the symptoms that we see in those patients, either they can be from the lack of the end product. So an albino person, he has a white skin, white eyes, because they don't have the melanin. So there is a lack of the end product. Or in some other cases, because of accumulation of a harmful metabolite. So instead of making one good metabolite that the body needs, then there will be accumulation of a bad metabolite. And we'll see examples of these diseases later in Mole 1 and 2. Okay, so usually how do we treat these pathways, this inborn error of metabolism? In most cases, we try to avoid, like if there is a missing enzyme in the metabolism of a certain nutrient, we can avoid giving this offending nutrients. Like if a patient will study with Dr. Ahmed, I think, diseases in the metabolism of fructose and galactose. So if you avoid, giving galactose, then the patients will be fine. So you avoid giving the offending nutrients. This is one way to treat these metabolic diseases. You will also see with, I think with Dr. Raja, about the storage disease. So we'll have the glycogen storage disease. What do we mean by storage disease? We have accumulation of molecules. So in the case of glycogen storage disease, you have accumulation of glycogen. Again, because of deficiencies in the enzymes needed to break down glycogen. In Mole 2, you will see lysosomal enzyme deficiencies, and you will understand more about this inborn error of amino acid metabolism. You should remember that these inborn error of metabolism, they are very rare. They occur 1 in 10,000 in usual population, like the American population, but they could be like in here because we have high rate of consanguinity, marriage between families. Some inborn error of metabolism, they could be more common. These diseases are usually recessive, so you need to inherit 2 copies of the faulty gene to have the disease. And heterozygous, usually they are healthy because you still have 1 copy of the gene making the enzyme and having the pathway happen. Okay, so this is an example of a disease that we've studied, I think, in Mole 2. This is in the purine metabolism. So in the breakdown of purine, there's 1 enzyme only that is missing. As a result, we have the severe cases, severe disease called Lesch-Nyhan syndrome. So what happened in Lesch-Nyhan syndrome? These patients, they will self-mutilate. So they will bite their lips and they bite their fingers off. So they will have severe pain also from inflammation in the joint because they have accumulation of uric acid, and they can have problem with the kidneys because from stone formation. So all of this because 1 enzyme here, which is the HGPRT enzyme that we'll study in Mole 2, is missing because of a genetic mutation. This is an example of inborn error of metabolism, and we'll see lots of them in biochemistry.